Greetings to you all. I am Jeremiah Paul, currently pursuing my final year BTech in biotechnology from the batch of 2023. Today, I would like to have give a very short presentation on my dissertation work of metagenomic analysis of human gut microbiome. I am currently doing my work at School of Life Sciences, University of Hyderabad under Dr. Vivek, and my internal guide is Rajeshri Yadaval. Coming to the introduction, I would like to briefly talk about the human gut microbiome. Human gut microbiome is the cohort of the body region right from your stomach to your intestines. This human gut microbiome harvests millions of microorganisms in every human individual, and these microorganisms play different roles like expressing for genes, creating pathways, and all. And one study stated that there are uh, around 3.3 million genes present just in the human gut microbiome. So my aim was to work on one class of these genes. So going ahead, what exactly is antimicrobial resistance? As I was mentioning previously, there are genes coding for antimicrobial resistance. What exactly is my antimicrobial resistance in the first place? It is the ability or the survival mechanism of a bacteria to overcome a particular drug or a drug class to show resistance and survive the attack of uh, different drugs. So this antimicrobial resistance is a negative thing for human gut because if a drug cannot work on your human body, it is as effective as not taking this drug. And there are hundreds and thousands of genes that are coding for this antimicrobial resistance, which I will be working on in my project. And coming to another interesting topic of horizontal gene transfer. So, horizontal gene transfer or HGT is the transfer of genes from one bacterial species to another bacterial species. So, this is a very common property of human gut or any bacterial environment of different bacteria living together in the same region. So going ahead with my aim of this project, it is to metagenomically analyze the human gut microbiome for antimicrobial resistant genes in Indian population. And my objectives were to detect the AMR genes in Indian population, check their trends and their resistance patterns, and to identify novel AMR genes present from this human gut microbiome of all the samples, and identify the causes of horizontal gene transfer or HGT in a population as to which organism is causing resistance in another organism. So going ahead with my pipeline of my methodology is the first thing is data acquisition. Data acquisition is gathering data or collecting data from reference websites such as NCBI where I take tons of data and analyze it. And the second step is quality check, that is to analyze and check the quality of all the raw reads that I get. And the third step is the trimming and cleaning of data, which is usually done to process those reads after the quality check to remove any sort of impurities, human contamination, or any sort of thing. So alignment of sequences is to align them against any reference database to see if there are any particular organisms present. I've referenced it against the human reference GRC38 to remove any human contamination from the gut microbiome samples. Since my microbiome samples are obtained from the stool samples, it is preferably not to have any human contamination. That is why I aligned it versus the human database and I've removed the contamination. And the fifth step is the assembly of sequences. Assembly of sequences is to combine all my reads into one large assembly file. And six is species genome bins. Species genome bins are nothing but small reconstructed bins which are made or which are created from the entire assembled sequence to give separate bins and separate organisms. So each bin represents an organism. And then seventhly, it is phylogenetic classification of bins, where I've given the full taxonomic classification for each bin. And eighthly, it is AMR gene detection. That is, I give it to a tool named as RGI, where it detects the resistant genes present in them. Ninthly, it is detection of the horizontal gene transfer. Tenthly, finally, detection of novel MR genes. So this is a map of all the samples that I've taken. I've taken about 215 samples from across the country to analyze. Right from the north to the south, I've taken samples from across the country to get a diverse view of this AMR genes or the microbiome in general. So I've taken WGS, that is whole genome sequence data, and every publicly available whole genome sequence data in India I've taken for my analysis, counting up to 215 samples. So let us skip quickly to the results part where I would like to discuss it briefly. This is the results where I got the phylogenetic classification of these bins. If you can see clearly from different phyla and different families, and this is the full taxonomic classification which will help me identify my reconstructed species genome bins. So the AMR gene. So this is a very interesting tool named as RGI, where it gives me all the AMR genes. These are all the AMR genes present in one bin, that is in Preventella copri, which is the most abundant bin in any human individual from an Indian background. Uh, and the AMR genes and the uh, it gives the gene families where they belong to, such as PMR phosphatase, VAN-W, OXA, beta-1,4, lactamase, or these are all the gene families uh, that code for these AMR genes, and the drug classes which they show resistance against. If you can see like tetracycline or phenol, disinfectant, antibiotics and antibodies, and penem, these are all drug classes which these genes that I've shown in the previous slide show resistance against. So once analyzing all 215 samples, I've made an histogram of all the data across all the human samples. So this are two interesting histogram plots. One is species resistance based on drug class. So if you can see, there are 96 bins that I have reconstructed previously. So there are glycopeptides, that is 59 bins out of 96, and tetracycline and fluoroquinone and disinfecting agents. So these are the most commonly present resistance patterns. So if you take if it you potentially take a drug from the family of a glycopeptide, it is pretty much sure you're not gonna have any effect on your body, because like 70% of your gut shows a resistance to it. So for it to be very effective, it's a very negligible chance. And also in plot of the resistance mechanism of genes, which is antibiotic target alteration and antibiotic efflux, which are the most two common methods for all genes. So what happens with this antibiotic target alteration is that the genes or the bacterial species do not all recognize the genes properly, so they alter the target for them to be even effective. And going ahead with this horizontal gene transfer, where I've tried to prove a hypothesis of an wet lab experiment, where they said the horizontal gene transfer occurs only between species of the same phyla. So if you can see, this is bacteroids containing Preventol, as I was mentioning earlier, and all different species from the same phyla of bacteroids. And this is from Probacteria, and as you can see, there are like every map plot over here is the transfer of genes. So every mapping between two species, if you can see this Preventola, and if you can see follow this blue line, it goes to GGB1495. That means from GG95 the genes are getting transferred to Preventola, and every line is indication of a horizontal gene transfer. So I was trying to prove the hypothesis of whether these genes can transfer only between the same phyla. And I've compared bacterioids and probacteria and put them into the same HGT plot. And if you can see the first five, that is the left end of it, all belong to the probacteria. And if you can see very clearly, the HGT occurs only between these five species, and the bottom five belong to bacterioids, and the HGT occurs only between them. So there is no computationally detected transfer of gene from bacterioids to probacteria, which you can hypothesize and confirm that the transfer of HGT can will occur only between the same species and not uh, within the same phyla and not cross phyla. So that is one conclusion that I could make of it. And though how much we conclude and how much we computationally analyze, it can be only very limited, because at the end of the day it has to be done in the proper lab facility. I can provide them the data for it, but the validation needs to be done from a wet lab standpoint of view. Like, I would like to just conclude by saying the resistance in the human gut of Indian samples is very high. I, being an Indian, I should be very cautious of the antibiotics I'm taking, because once I keep taking it, I will start developing resistance against it as well. So I would just uh, suggest all of you listening to this presentation, be aware of what you're taking into your body, because over time it's not going to work. One classical example of it is paracetamol. Nobody has paracetamol anymore in 2023, because the human body started showing resistance against it, and it really isn't that effective anymore. So people are asking you to have a higher dosage of the same drug, that is Dolo 650. So that is one classical example of uh, people developing resistance to a drug. So try to avoid drugs as much as you can. If you are taking it, take it in a cautious and an approach, so that you don't overcome and have resistance towards this. And with time, I would really hope that it comes down with people being more aware of their health situation. So thank you and have a great day.